Jack Gilbert, thank you very, very much for agreeing to have a chat with me, all the way from sunny or not so sunny San Diego. Um, I kind of had a bit of difficulty in trying to figure out a theme for what I talk to you about because your research is just so wildly all over the place. Still relating to the microbiome, but the microbiome of everything. In fact, I watched one of your talks where you described yourself as a pediatric oceanographer at one point to describe the kind of breadth of the research you do in the field. So maybe we can just start off and you can give a little introduction to how you got to where you are and how did you start off and how did you get to this kind of crazy place you're in now.
I would say the you could sum it up as academic ADHD, right? I love everything, and I have so many questions I want to ask, and I get it's weird. As soon as we get a grant to work on something, I'm less excited about actually doing it. I love the ideas. I love creating research plans to develop those ideas. But my background's kind of weird. I worked at the I did my undergraduate at King's College in London, and then ended up working for the Natural History Museum down in
No
Kensington
way.
for a long time, about a year and a bit, cataloging their butterfly distribution patterns. So, like
Amazing.
cataloging their collections and putting them into a computational platform to look at biodiversity in Africa. And I absolutely loved that. I thought I was going to be an entomologist for my whole life. And then weirdly, out of the blue, on my lunchtime one day, I can't remember when it was, somebody emailed me and goes, "Your name has come to my attention. We would like to offer you the opportunity to do a PhD at Nottingham University, but it means you'd have to go and live in Antarctica for 2 years." And I was like, "What?" I was like, "Who is this?" And so I followed it up, and lo and behold, I don't know how my name got into their lexicon, but they offered me the PhD position,
Yeah.
and so I spent 2 years working on microbes. And I told them I had no microbiology background really, apart from an undergraduate course. But they said you can learn that. You can learn that. It'll be fine.
Yeah.
And that you can learn that it will be fine has pretty much been the running theme for my entire career.
Oh, of course. Yeah.
We'll
As with
figure
lots
it
of us
out as we go.
scientists. Yeah, yeah.
Exactly.
We'll figure it out.
Build the plane while flying it. It's absolutely fun.
Exactly.
And so it's been a huge amount of fun. I bounced around from microbiology in Antarctica. Then I went to Canada to do a postdoc where I had to learn proteomics and protein characterization, X-ray crystallography, that kind of stuff. I came back to England to work at Plymouth Marine Laboratory for about 5 years until 2010, where we were doing marine microbiology. And then I went to University of Chicago and Argonne National Labs in Chicago. I didn't even know where Chicago was. That's how ignorant I was of US geography.
You only
I
knew the
went there for
UK and
9 years.
Antarctica, but you didn't know
Right.
the geography of the US.
I was like, Chicago is somewhere in the middle,
Somewhere, yeah.
that's about as
Yeah.
much as I did. Worked there for 9 years. And then in 2019, I came over here for, my midlife crisis retirement into the
Wow.
sunny
So you've
clime
gone
so I
from
can surf.
butterflies to oceans and now to humans somewhat, which we'll get on to. I didn't actually know about that Antarctica a bit. Tell us about what that was like and what you'd kind of do day to day for science and what was it like living
Yeah.
there?
It was crazy. It was a privilege. It was me and 19 Australian guys who were part of the Australian Antarctic Research Division. And it was only men because they only one woman applied that year. So you can't, due to Australian regulations, uh, for gender reasons, you're not allowed. You have to have two women there because for the prolonged period of time, women need support of other women. That's what we were told. I don't know whether that would hold up under current gender regulations, but so it was very isolating from that perspective. I was very young. I was 22 when I went down, and yeah, it was a crazy experience. And my basic role there was to find bacteria that bind onto ice, and stop it from growing. Right, so these are ice inhibition bacteria.
Mm.
And that's a beautiful story. I actually got to go back to the lab which I worked on during that period. I got to go back and see their work because my supervisor was retiring. And it's funny. 20 years on, they've done some incredible things with the science. And they found that some of these bacteria bind onto ice, but they also using this appendage, but they also bind on the same appendage to a diatom. And if 6 of these bacteria bind onto a diatom, they can use their flagella to swim the diatom up to the surface of a lake, bind onto the ice on the surface where there's light and nutrient-rich water to feed the diatom. The diatom then produces sugars, which they consume. I mean,
No
how
way.
insane is that? That's so
No
cool.
way.
These
That's
bacteria
amazing.
hijack their own power plant facility and then take up to the top of the lake. And that's what blew me away is it took 20 years to uncover this, right? And I just started it by finding this bacteria in this lake.
Right.
And and you realize you're just part of a cog in a machine in academia, right? And
Yeah, exactly.
there
Yeah.
were
Yeah.
15 other postdocs and graduate students, galore followed me and did all this incredible work. And I love that. I love being part of a larger story. I never really
Amazing.
wanted to be, the leader of XYZ. I always wanted to be part of a larger story, a collaborator that can build up this amazing body of knowledge.
Right.
So I guess, yeah, long way of saying I love doing everything and I love being part of everything and
Yeah.
collaborating on everything. And it's stood me in good stead so far.
Well, you say that you love only being a part of it, but really you were a leader in one of the big kind of things you're known for is the Earth Microbiome project, and you led this to try and catalog, living microbes in huge number of environments all around the world. You were not to try and blow your trumpet or something like that, but were like Darwin trying to systematically catalog microbes which we don't know enough about in all kind of regions of the world. So tell us a little bit about that, and how close are we to getting to where Darwin was in his day?
Well, I think I always say if Darwin was alive today, he would have been a microbiome scientist, right? And
Yeah, yeah.
for me, that holds true because microbes and their potential to evolve and adapt and and manage the environments around them, they're phenomenal, right? They are just incredible. What he was inferring from finches and earthworms, we are observing in microbes on a regular basis. And so that was awesome. Yeah, we started the Earth Microbiome Project in 2010, and there were about 20 or 30 of us locked in a hotel room above Salt Lake City and doing some pretty heavy drinking, but pretty cool theorizing, about what could you do if you could sequence everything, right? And out of that came Rob Knight, who's obviously the super 50,000-pound gorilla in microbiome science, and one of my close friends and colleagues here at UCSD, and Janet Jansen, who's up at Pacific Northwest National Lab. And so they really, Rob had all of the expertise and technical experience. I was just a wonderful cheerleader and absolutely loved doing it and had some funds to help kickstart the program. So for me, it was always a part of being part of that group and the resulting 450 other scientists that we collaborated with who also played major roles, right? So not so much of a leader, more of just a herder of cats in getting the program to actually work properly. But it was a phenomenal, for me, a phenomenal honor to be part of such a large group of people doing some pretty cool science. And it's still ongoing, right? We in 2017, we published the first paper with 27,000 samples processed. We're now up to about 160,000 samples processed, and data is available. It's publicly available on multiple forums.
Right.
And people can now use that. They can take their microbiome data from their study and put it in context to the
Mm,
global microbiome. And that's important if you want to understand ecological trends.
mm.
But when will we be done? Never, probably. Because microbes are evolving. And our main goal is to understand how they interact with
Mhm.
each other, how they manipulate their environment, and what that actually means for the globe, and how the globe is responding to the current crises,
Wow.
such as climate change, which are affecting it.
Yeah.
And so, yeah, we're in a continuous discovery pipeline. And I think
Brilliant.
Darwin would approve of that, right? I don't
Yeah,
think
I
he ever
definitely
wanted
think
to
so.
reach a finality.
So give us a few examples of the kinds of cool places that you have data from in the Earth Microbiome Project. Where is the equivalent of Darwin's Galapagos Islands, where you have this huge diversity that you didn't know much about before? What are the kind of cool places?
I mean, this is what's fascinating for me. And we did a lot of work on this in 2008 to 2011, approximately, so about a decade ago now. And we actually found out one of the most diverse places on the planet. If you look at it from the data, is actually the English Channel, which makes no sense. It's totally irrational. But when we sequence really deeply in the English Channel, you
Really?
find all of the microbes, more than 60% of all of the bacteria we observed in any other environment in the ocean globally, right? So that suggests that we shouldn't think more of, it's not like the Galapagos Islands is the most, diverse place in the world. We don't really have that, simulacra in microbiome science because microbes are distributing around the world so regularly. In terms of numbers, the most diverse places are soil and sediment, right? Because they're the ones that have the most niche structure. Most little individual niches on, within say a gram of soil. There's so many surfaces which bacteria can colonize. Interestingly, for any gram of soil, only about 20% of the surfaces of the soil grains are actually colonized by bacteria. 80% is just a blank desert with no biological material. But they are, it's a phenomenally rich, complex environment. But because microbes can move around the world, maybe bass specking was potentially correct, and everything is everywhere, and the environment selects. I don't think that's true in any given moment in any given day. But if you look at geological time, or at least thousands of years, then I think it's very true that we are living in a microbial world. They permeate everything, and they're just adapting to whatever comes along, be it a chunk of ape or buildings that we are now creating our built environment, which is a massive selective pressure against microbial evolution and changing the paradigm for many of the microbes that we've come to know and love or hate.
That's amazing. Yeah. And I want to come on to that into the buildings next. But, say you did go back and sample some of these places. I mean, we all know that microbes evolve really, really fast, much quicker than we as humans evolve. So, how long would it take for you to go back and see completely new species if you were to go back to some of these places that,
Oh,
obviously that depends on the environment and climate change and things like that. But, how fast are these actual changes happening to these kind of communities that you're sampling?
I would argue we'd have to define the parameters of
Well,
your
yeah,
investigation,
yeah,
right?
exactly.
What's the species?
Well, there you go. Yeah, it depends. Yeah.
Like if you take say a hydrothermal vent, we see a very very selective environment, high temperature, high pressure, zero light, lots of sulfur, so lots of sulfate reducing potential, right? Um, and are the organisms that we saw there 10 years ago the same as the organisms we
Mm,
see now? Well, most of those vents don't last 10 years,
right.
so it's hard to say, isn't it? But I would argue, from what we can tell, if you look at strain level or genotype level organisms, right? This is a microbe with a unique genotype. Not just one or two SNPs, but a single nucleotide polymorphisms, but enough genetic manipulation to say, yeah, that can do something different. It has a different functional phenotype, maybe very similar to its strain siblings, but it has the potential to do something different. Well, we find those cropping up all the time. When we looked in 2018, 2017, we started this project in 2010, but we were looking at the oil pollution that came out of the Macondo wellhead
Mm,
explosion in the Gulf of Mexico in 2010. And we found that the genetic diversity of the microbes in the sediment that were responding to the Macondo oil head explosion was phenomenal. A lot of
really?
these bacteria, the genotypes, were found all over the Gulf of Mexico. We could find
No
them in
way.
sediments that had no oil associated with them whatsoever, but they were very low in abundance. And then around the oil head, they became super abundant. And
Really?
each one had a unique adaptation to degrading one type of polyaromatic hydrocarbon, right? So
Really?
they were each one was nibbling away at different parts of the crude oil which had seeped out. And that to me was crazy. These guys are hanging around waiting for something to happen. Well, each one of them is also undergoing genetic mutation and therefore has the potential to alter its phenotype in response to the world around it. And that's the beauty of microbes, as you said.
That's phenomenal. That's brilliant. So you moved on then, you kind of had looking at the kind of the bigger environment all around the world, and then you kind of wanted to start looking at closer to home, and honing things in a bit. And you've published some really interesting stuff on the microbiomes within the buildings around us in various different settings. One of which is in hospitals. And I know there's some really interesting anecdotes and studies showing how the microbiome of the hospital buildings can actually influence infection rates in patients. And tell us a little bit about that, and why the kind of microbiomes of hospital buildings might actually be important for the humans within those buildings.
Yeah, I mean, the key takeaway from all of our studies, we published a study in 2017 that basically demonstrated that, what we'd found before, that when a human being enters a room, they shed so many bacteria that their microbes, the ones that can survive in a building, which are very, very few. 99% of bacteria that leave your body die, right? But the 1% that do survive, they are your microbes, and they colonize the space that you're in. Each one of us is emitting 38 million bacterial cells an hour, according to Jordan Petchier at Yale, who did some phenomenal work in that space. But the outcome is, the patients enter a room and their microbes leave their body. Now, what happens to the ones that do survive is very interesting. What we found through extensive metagenomic analysis of over 100 samples, and then reconstructing the evolutionary selective pressure upon operons that were associated with antibiotic resistance, is that the bacteria that were in the room were being selected for antibiotic resistance, but not by the antibiotics the patient was getting. And that was really interesting to us, right? So it didn't matter what antibiotic the patient was receiving, the organisms were being selected for antibiotic resistance genes that had no relationship to that antibiotic. And we hypothesize, based on the evidence that we received from there and recent studies that we've been doing that haven't been published, that the bacterial operons that are associated with antibiotic resistance are also associated with survival in extreme environments. Right. So you've got things like Staph aureus, Strep pneumoniae, Pseudomonas aeruginosa, and they're leaving the bodies of these patients, ending up on cold, dry, harsh surfaces where there's virtually no moisture. And they're surviving, but they're surviving on the edge of survival. And so the selective pressure of that survival environment is also enabling these organisms that just so happen to contain antibiotic resistance genes on those operons to survive and thrive. Right? Well, thrive is hard, but they're definitely surviving.
Anyway. Yeah.
And so the potential for those to recolonize people who are susceptible is great, right? And so we're doing a double whammy. We're working with people who develop phages to kill off the bugs inside the patient, but we're also working on on strategies to alter the building environment, potentially by adding bacteria back in, Bacillus subtilis spores, for example, to just saturate the environment with good bacteria, or benign bacteria, that can outcompete these bugs that can survive when they leave our body in that space. So it's biological control in a hospital. Yeah, and we think that's much better than sterilizing everything. Because if you sterilize everything, the patient doesn't stop emitting bacteria into the environment. So if you sterilize a surface, 2 or 3 minutes later, the patient's bacteria have recolonized it.
Yeah, and so I think there's evidence of that, isn't there? I'd read some interesting anecdotes, at least anyway, that hospitals that had installed this kind of antimicrobial flooring, hoping that it would reduce infection rates, but actually it increased infection rates because it kind of led to those bacteria.
Well, it can select. Yeah, the bugs, as you pointed out earlier, the bugs find a way.
Yeah,
it's like very Michael Crichton. The life will find a way. But microbes do. They find a way around what we're trying to do. I mean, I often say, look, if you really, really want to be super sterile, the best thing to do would be cover every surface in copper, right? It's very hard for microbes to evolve a mechanism to survive or resist, copper oxidation from the divalent metal ion getting into it should wipe out everything, right? But covering the entire surface of a hospital in copper would be incredibly expensive, and almost and potentially, and may, you don't know, but it may lead to resistance. It would be crazy if it did. But that's the exciting piece. So we need other solutions. Things are working quite well, but we need other solutions to these problems.
So tell us the practicalities of that. How do you fill a hospital potentially with these kind of healthy or kind of non-disease causing microbes in order to do that? Are you spraying it around the place? Are
Yeah.
you really?
Well, I mean, researchers are already doing this. I mean, you can buy products on the market, on Amazon, whatever your favorite uh, online retailer is, that contain bacillus spores in them. So you wash your home with bacillus spores if you want. These are just standard cleaning products, spray bottles, floor cleaners, the issue is we just don't have a huge amount of evidence to suggest it actually does anything.
Hmm.
Now, there is a group in Italy and one group in Florida that have done limited clinical studies and demonstrated that the addition of these bacillus spores compared to standard cleaning tends to reduce the abundance of antibiotic resistance genes in the environment,
Right.
but they're very small. Now, we're doing much larger studies, replicated at a much larger level to demonstrate its efficacy, we hope, right, as a potential strategy. But it's one in a box of arsenals. One that is interesting is we've also just recently demonstrated that SARS-CoV-2, like the flu virus, binds to the outside of some of these bacteria, which can survive in the outside of the body, right? So if you think if SARS-CoV-2 finds its way into someone's lungs, and that person has Staphylococcus aureus in there, and the SARS-CoV-2 can bind onto the outside of Staph aureus, then when both of those are released into the environment, the SARS-CoV-2 can survive longer outside the body on cold, dry surfaces because it's bound onto this bacteria, which can also survive, right? And then if that's re-inhaled, the bacteria finds a neat little way of getting back into the body because the SARS-CoV-2 can help the bacteria to invade human cells because the lock and key mechanism of the spike protein. But it can also help the virus because then the body goes into paraplegisms of immune response to deal with the Staph aureus and leaves the SARS-CoV-2 alone. And so there's a great researcher, Jason Roche, at St Jude's Hospital, who did this work in flu and demonstrated it quite effectively that flu transmission is much greater if ferrets have particular types of bacteria in their lung because of this transmission potential. And we're now demonstrating that in SARS-CoV-2. And so it may be that, if a SARS-CoV-2 Staph aureus conjugate lands in an environment flooded with bacillus spores, it may not survive and may not have the greater potential to cause transmission. So we think there may be benefits even to the current pandemic of doing this. And that's exactly what we're researching now.
Wow, amazing. So how does that work then? So we're kind of honing in on ourselves here. We started like Earth and hospital, and now we're coming a little closer to home in the home itself. I made you choke in your tea. Give you a minute.
Yeah, sorry about that.
No, you're all right. You're all right. It's the thought of all these spores and everything. So you've looked in the home as well. You've said that we're all emitting 30 million cells an hour, or whatever is it, when we go into a room. So how much of that then in a confined environment, like in a home, is actually transferred from individual to individual? So say I have a new roommate that comes in and lives with me, am I actually transferring that much? And how is that transfer happening? Is it airborne? Is it through surfaces? How much are we kind of
it's a really good question.
Microbes?
Yeah, when we look at say people that become roommates, there is a certain degree of microbial exchange. And we looked at this in Air Force cadets. So these are people who come from all over the country to a Air Force Academy. Sorry, that T really did a number on my my larynx.
It's all right.
T is very dangerous. And they came from all over the country. They stayed in, and you get these. They stay in a duplex room, right? So the room has like a dormitory in a university. It has 2 beds, 2 tables, and they use those rooms. But they all eat the same food because it's the military. They all do the same exercise regimen. They all have the same haircuts. They all wear the same clothing. It's very, very prescribed. And in that study, which was led by the military, we noticed that these young, very healthy people, tend to only exchange about 5% of their microbes. So they'll only become about 3 to 5% microbially similar, right? Otherwise, most of the microbes just mix in the environment.
Right.
We don't fundamentally understand the mechanism of that transmission. We hypothesize it's through that shared activity and then inhaling it. On your skin, any microbial contamination, let's call it that, that you pick up can easily be washed off with hand washing, right? And then
Mm.
your own microbiome will bubble up through the skin, the depths of your skin to the back to the surface. For example, if you and I shook hands, we did this with for NPR once with what's it called? The physicist who's the on the TV all the time for Cosmos, Neil deGrasse Tyson.
Oh
We
yeah,
had him
yeah.
shake hands with somebody, and then we swabbed their hands every minute for about 5 minutes. And you can see the microbial evolution of his microbiome coming back to his hand. There's a bit of contamination to begin with, and then it resurfaces, it restructures itself. And so there's always this if we clean someone's hand, their skin will come back to their own microbiome and eradicate whatever signature existed previously. But prolonged exposure will have a big impact. If those two people are physically interacting, that changes everything entirely.
Right.
Swapping saliva, as it were, and physical conjugation can have a dramatic effect, leading to a higher degree of similarity. But even married couples will retain unique microbial identities over decades of cohabitation. And so there is an element there that your microbial self stays who you are. You just swap a certain
Right.
amount.
Right.
But Rob Knight did a wonderful study once where he looked at couples living together that either had dogs or babies or nothing. And couples with a dog were more microbially similar to each other, i.e., they shared more of each other's bacteria than couples with a baby or couples without a dog.
Really?
So there's something else going on there that we can't quite tell.
Being
Maybe
passed through the
dogs,
dog somehow. Yeah.
right? Maybe they're super transmitters. Like a great vector for microbial transmission. We don't know, but it's that kind of understanding of how the microbes are transmitted. So something we it's very hard to
Right.
determine,
Yeah.
even in animal models, right? Because hard to track the movement of all of these
Mm,
microbes. We
mm,
use mathematical algorithms to do it, but they're not perfect.
right. That's interesting. So you kind of retain a lot of your own microbiome. So presumably there's something ingrained from earlier in life that forces you to kind of have your own microbiome, or maybe not earlier in life, just something about your body that brings it back. So what
But
do we know then?
you're an ecosystem, right? And your
Right.
immune system is kind of unique as well. And so the combination of having that, Darwin would be proud, right? That you are an island, you're colonized as an island, and whatever that starting colony is will
Right. Yeah.
shape how your ecosystem develops, and your immune system shapes that as well. So that partnership between immune system and microbiome is going to be continually shaping and structuring who you are. You can change that dramatically over your life through diet, antibiotic therapy, etc. But for the most part, it's always going to bounce back to
Mm,
who you were originally.
mm, amazing. And so if there is that transfer between people, do we know enough yet what the implications for that are? Say there is this 5% transfer you're saying, or more if you're kind of physically interactive with people. Is there any evidence to show that that is actually has any implications for health, or is that enough to influence this island or this host?
We don't have any evidence at all to suggest it influences health. There's lots of data to suggest that interacting with fellow siblings, for example, that have gone to school, can influence your immune response as a child. Fantastic work from Susan Lynch in University of California, San Francisco, and groups in Germany. I'm trying to remember her name. Oh, it's terrible because she's a really good friend of mine. But anyway,
Somebody else.
it'll come to
Yeah.
me later.
Yeah.
Have demonstrated quite effectively that children growing up in a household that have older siblings that go to school will have a stronger immune response
Mhm.
and are less likely to develop asthma or allergic disease than children who do not have older siblings that are at school. So there's that element that interacting with those children that have that other microbial exposure could be playing a role. But it's all circumstantial to a certain extent. We cannot unpick the mechanism that underlies that interaction. But the work we've done with the Amish and Hutterites, looking at asthma effects, has demonstrated that children interacting with furry animals, and to a certain extent, Susan Lynch's work with dogs and cats, children interacting with furry animals very early in life can have a profound effect on their immune response.
Mm.
So there's an element of that. If you grow up with a dog and you can physically interact with it under the age of 1, you'll have a 13% reduction in the likelihood of developing asthma. If
Right.
you grow up on a farm interacting with farm animals, you have a 50% reduction in the likelihood of developing asthma, which is phenomenal, right?
Right.
So maybe interacting with humans could be beneficial, but interacting with furry pets or furry farm animals, that might be even
Even
better,
better.
right?
Yeah.
So
Right.
worth thinking about.
And so one of these other kind of unique environments you've looked in, we know that if we have exposure to animals, we have exposure to other people, that's probably a good thing, kind of increasing this diversity. But there's some cases where people are, exposed to a limited environment, one of which I suppose is the International Space Station, which I think you've looked at as well. And that, I
Yeah.
suppose, is a really unique environment to look at because these people are in a confined space for 6 months, a year, with only, I think, 5 other people. And not any real exposure to the outside world and these other external organisms that you're talking about. So what happens there? We know there's long-term implications for health for these people. Maybe not long-term, but there is implications for when they come back.
Yeah.
Some of that to do with their changing microbiomes, or how much do their microbiomes change when they're up there?
We're trying to understand that, right? And it's really complicated. And the complication is these are really small groups of humans. And so we don't have any statistical power to actually assess what's going on. So all of the human studies actually on the International Space Station are really limited and really hard to actually infer anything from. However, our main interest is does the environment shape microbial evolution? And so, we currently we're sending up more materials up to the International Space Station a couple of months' time on a SpaceX resupply because we want to do time lapse observations of microbial evolution in that space. We already know that if we space fly isolates of fungi or bacteria and we bring them back down to Earth, they'll be more pathogenic than the same organism that just stays on Earth for the duration of that exposure, right? So, if we infect nematodes with those bacteria or fungi, more nematodes die if the organism has been space flown. We don't know why, though, right? We, however, hypothesize that the combined effect of being in this cold, dry, harsh environment maybe combined with more radiation exposure from cosmic rays and the sun, maybe also zero gravity, is somehow shaping these organisms' evolutionary process or their epigenetic process in a way that has an impact upon their pathogenicity or virulence. But we are at really early stages of trying to figure that out. So our main issue is, say you got an astronaut and she has to fly to Mars, and she's going to be there for 6 months in a spacecraft, right, with maybe 3 other people. But she's going to be there also with, 30 to 40 trillion bacteria in her body and 30, 40 trillion in the other ones. And those bacteria are leaving her body, ending up in this environment that is shaping pathogenicity. If she does become susceptible to an infection, is there the opportunity for these built environment microbes in the spacecraft to reinfect her because of this heightened virulence activation, whatever's driving it, right? And so we're really interested in understanding, is it possible to ameliorate that? And so maybe taking our bacillus strategy for hospitals and imposing it into spacecraft could be a way of just preventing that from happening by preventing these organisms from ever establishing in the International Space Station. But then the question is, does bacillus mutate into some horrifying, monster spacecraft bacteria? I'm currently having those epidemiological fights with NASA to say, like, no, it's going to be fine. Well, how do you know? Well, I don't know, but it's a problem.
There's definitely going to be a movie made about that. This scientist who says everything's going to be fine, and suddenly it all.
It's all going to be fine.
It's
Don't
all
worry.
fine. No.
We've got space zombies and everything's gone
Well,
crazy
exactly.
on Mars.
Yeah. Maybe that. It is worrying. I know there's talk of this kind of like colonization of Mars and all these other places, and really that could be our downfall. Is the microbes that we bring up with us? Rather than.
Right.
Rather than us being the architects of our own downfall through, I don't know.
Well, that's why I think we need to take dogs into space, right? Laika was the first one; she didn't survive. Well, we need dogs in space with the people. Why can't we have dogs in the International Space Station? They'd love it, right? The poop cleanup might be a problem.
Floating around.
When we go to Mars, we should have dogs there.
Yeah, that'd be a great idea. Yeah.
Yeah, I think it's
Brilliant.
awesome.
All right, well, you can suggest that to NASA now with one of your next
Oh, I already have. They
next
poo-pooed
meetings.
the idea.
I think we'll leave it at that. I don't know. What do you see? I was about to kind of ask you what is the future, but I feel like we're talking about the future already with all these space things. Like, what are you excited about next? You've done Antarctica, you've done the Earth microbiome, you've done space. What
I'll tell you
what
the
do you
two
see?
things I'm most excited about, right? As you can tell, I generally get very excited about all of it, right? But what I'm really want to do, we've done lot of observation for the last 10 years. We built some phenomenal hypotheses. The two things I'm most interested in doing are precision therapies for humans, right? I really want to get to the stage where we've identified, say, bacteria release this chemical, this metabolite into the bloodstream of humans. It can be an indicator of disease, but could it also be a therapy? We found certain bacteria release certain metabolites into the serum, which actually alter the allergic response of immune cells in children's lungs and prevent them from developing asthma. It's a hypothesis. We've got mouse work that suggests it. I really want to get to the stage where we can test that in a clinical trial. And I'm maybe 1 to 2 years away from doing that. But if I take this metabolite and I can inject it into children's bloodstreams early in life who are susceptible to developing asthma, can I reduce their chances of developing it? 12% of the US population can develop asthma in their lifetime. That's insane,
And
right?
growing, and growing probably.
It's growing, and it's a debilitating disease that we may be able to prevent. So I'm really keen on doing that, and many other diseases. But come back to environments. We're working extensively now with groups like one of my colleagues, Jeff Bowman and Sarah Allard at Scripps Institution of Oceanography, and colleagues around the world as part of a new consortium we've built up, like an Earth microbiome focused on mangroves. Right, mangroves suck up so much more carbon, 5 to 6 times the amount of carbon as terrestrial forests, and they're nurseries for fish, so they're important for reestablishing fish stocks, and they clean up all the pollution coming off the land, so they prevent the degradation of coral reefs.
Wow.
These are incredibly important environments, and so we're working to build microbial probiotics, so specific microbial organisms that can be placed into the root systems of planted mangrove trees to promote their growth, to ameliorate and remove pollution, so we can actually re-establish mangrove forests around the world. And those are just two examples, right? We're translating our science now. And that's what I'm most interested in doing. That's the legacy I'd like to leave when I shuffle off this mortal coil, is that we've actually developed some therapies for the earth and for humans that will actually have lasting impacts on the health of our planet and our populations.
Wow.